Hej och välkomna. Vi ska prata med Bioinvents vd Martin Welschof. Good morning, Martin. How are you?
Good morning. I'm very well. How are you?
I'm perfect, thank you. I would like to ask you, you just reported your third quarter. Could you perhaps tell us some of your highlights from the third quarter?
Absolutely. So, as everybody probably knows by now, so we have a broad portfolio, and actually we have delivered across the portfolio during the last couple of months, basically. So we had, have now demonstrated strong single-agent activity for our lead anti-TNF receptor 2 program. And then also we moved ahead strongly with our other lead, targeting Fc gamma R2b. And there actually we had additional data that we have disclosed now in this report. The market might remember that we had good data at EA, but that was preliminary, where we saw strong, complete, and partial responses. And basically we could confirm the trend now in a larger patient population. And that was also mainly the reason why we pushed ahead now with the subQ in the combination, separate combination with acalabrutinib and rituximab. And there we also have started recruiting already. And then, of course, all the other programs are also going full steam. And then we also disclosed a quite busy next year. So we still have 3 milestones outstanding for this year. So by the end, we'll update on the 1808 single-agent activity. We'll have the first data set for 1206 in combination with acalabrutinib and rituximab. And then also, I think very interestingly, our second anti-TNF receptor 2 program, 1910, that will report the first clinical data as a single agent. And that brings us also in a very good position around TNF receptors 2. I think we have the strongest package to date in comparison to our competition.
And you have quite some, you have many collaborations with different partners. Could you perhaps tell us a bit about those partnerships?
Yes, we have a number of collaborations. So the company has basically validated the platform that we're also using to generate clinical candidates with a number of pharma companies, such as Mitsubishi, Takeda, Daiichi, just to mention a few. And those programs are running also quite strong. So this is basically a secondary portfolio that we have with those partners. Clinical trials are now either phase 1 or phase 2. And I think the first 2 we are moving closer to phase 3, which is quite interesting. And of course, for those programs, there was an upfront payment. There have been preclinical and clinical milestones, and there will be commercial milestones and tier royalties. And then we have partnerships based on our proprietary programs, and one is 1206 that we have partnered with CASI for China, including Hong Kong, Taiwan, and Macau.
Yeah, and with the CASI, it's only collaboration around the Chinese, the Asian market. But what about the American market, for example? Do you have a different partnership for that, or can you add some new partnership to that?
Yes, so basically the CASI collaboration is the only collaboration, and it's not Asian, just to make it very clear. So it's only China, Hong Kong, Macau, and Taiwan. So it's what you would call, maybe not politically correct, Greater China. And all the other programs are basically unpartnered. So talking specifically about 1206. So we still, of course, have the European rights or all other rights for the rest of the world. And the only other collaboration that we have, or collaborations, I should say, is clinical supply and clinical development collaborations. So we have 5 with Merck and 1 with AstraZeneca, which I think is also very strong because they don't have any rights, but they support us in the development of those programs with supply either of pembrolizumab or acalabrutinib, and of course help us also with insights how to best develop our compound in collaboration or in combination with their compound.
And what differentiates? Why would you choose, as a partner, why would you choose Bioinvent? What do you add? What is your specialty?
Our specialty is really to identify and develop antibodies for immuno-oncology, and we're specifically focusing on the tumor microenvironment, which is very interesting in the sense of immuno-oncology. And I think besides the antibody technology, and that's not only target discovery and then antibody discovery and manufacturing, We are quite integrated as a company. We can produce and formulate our own drugs. And then we also do clinical development all under one roof. And I think then we have one of the companies with the largest biological experience around the tumor microenvironment, because that's very important that you not only can generate your compounds in a straightforward and high quality way, but also that you really understand what you're trying to target and to tackle. And I think this combination is unique for Bioinvent.
Are you also looking into ADCs?
Well, we have the potential to do that. I think currently our focus is clearly on naked antibodies. And keep in mind, if a naked antibody can do the job, it's much preferable from a safety perspective, but also from a cost of good perspective, because it's much cheaper, much more straightforward to produce. And also ADCs, at least the ones that are currently in development, have problems sometimes with toxicity, which we don't have. So all our programs are completely safe, even though they show good efficacy. And I think that's always the preference. But in one or the other case, ADCs might sense, and then we might consider that as well.
And regarding toxicities, I'm thinking about the IV administration that you had in BI-1206. You went more into subcutaneous with that. Why? Is that because of treatment emergent adverse events, or is it something else that made you go into this?
There were really 2 points. So one was with the IV, we had mild infusion-related reactions, the same as you would see if you treat with rituximab, which is approved and a completely safe drugs and other antibodies, and didn't have any clinical manifestations. But we thought since we can do it because we have the capabilities in-house, we try sub-Q, and we now have a perfectly safe sub-Q formulation. All the infusion-related reactions are gone. And on top of it, and that was the other point, it's much more convenient, because if you do sub-Q, this takes 5 minutes compared to IV, which could be at least an hour or maybe even 2 hours.
Understand. I'm also thinking of the BI-1808. Maybe you could say something about it. When can we expect data? I think it's at the end of the year, or how is that? When?
Yes, so BI-1808. So there I would like to talk a little bit about TNF receptor 2. And TNF receptor 2 is a target that we identified when we screened directly on ovarian cancer with our first technology platform, which is very unique since it allows us to screen directly on patient material. And we found this in ovarian cancer patients as a very interesting targets. And then we started to work on it and basically identified several hundred antibodies. Out of those, we selected 2. One is the lead 1808, and the other one is the second candidate, which is 1910, with 2 very distinct, uniquely different mode of actions, both translating into immuno-oncology therapeutic agents. And 1808 now is more advanced. So there we already have demonstrated strong single-agent activity in patients that have been heavily pretreated. So in one case, for instance, a GIST patient, this patient received 12 prior lines of treatment, didn't have any more treatment options, and then we came with 1808 single agent, and this patient responded very well. So I think this is very, very strong data. At the same time point, we developed this also in combination with pembrolizumab, because when you think about cancer therapy, at the end, this is all about combination therapy, and the combination data we'll have at some time point next year. And then what we also disclosed, we'll combine this also now with chemotherapy. And again, then we try 1808 in all the combination that would make sense, in addition to the single agent therapeutic arm that we're running. And that will give, I think, a very, very interesting data set. And I think in total, with 1808 and 1910, for which we also will get the first single agent data by the end of the year, we have the strongest package around a very unique, interesting target that's coming up now. We're well ahead of the competition. And as I said, have the most comprehensive package around that target with 1808 and 1910. So it's very cool.
Interesting. You have cash runway until first quarter of 2026, right? still
Correct.
on track?
Correct.
Yeah.
So we're
And you're gonna
yeah, absolutely on track.
Yeah, and you will also attend Jefferies and JP Morgan in January, right?
Absolutely.
Yeah. Okay.
Absolutely.
I'm wishing you all the best, Martin, and thank you very much for saying hi and talking to us here at the EFN. Thank you.
Thank you.